feel my face. I mean, I can touch it, but I can't feel it inside.
Ladies and gentlemen, welcome back to the True Life Podcast. We are here with Jachym. Am I pronouncing it right, Jachym?
Yeah, it's Jachym.
Jachym. Okay, nice. He. I should have the bio written down right here. Jachym, would you be so kind as to maybe give people a short introduction into who you are, what you're passionate about, and maybe April 19th, what you've been working on?
All right. So I am originally a medicinal chemist, so that's how I started my academic career at the University of Chemistry and Technology in Prague. And I studied drug production and synthesis. So it's really kind of building on my interest in chemistry throughout my life. And this vision that I want to, discover, invent a new drug that's going to help a lot of people. And I started working in the forensic laboratory of biologically active substances at my uni. And this is really a laboratory focused on various psychoactive substances and studying them, analyzing them, and synthesizing them for different purposes for research. And I initially worked on cannabinoids when I was in my undergrad, and didn't turn out to be really fruitful line of research that I was doing, and I wanted to do psychedelics because I was really interested in them, and there was this really amazing new research about the mechanisms of action coming out just at the time. And my supervisors at the laboratory were so kind to give me the option to come up with my own topic for my diploma thesis, if it's any good. So I was looking through these different papers that were coming out, and I formed a hypothesis about really cool kind of compounds that could have even like higher or better way of increasing neuroplasticity within the brain while also having neuroprotective effects. And I decided to really pursue that. So really design the compounds so that this profile of the hypothesis, and that's what got me into drug design. And I studied drug design at UCL in London also, and then went on to try and synthesize the compounds that I've designed. And it really is this now hypothesis about this pharmacologically unique kind of compounds that I am trying to basically develop into a new kind of psychedelic therapy, psychedelic-like therapy. And the focus is now that these could be perfect for indications in neurodegeneration and traumatic brain injuries, because they would allow even better protection of the brain and better prevent neurodegeneration and aid the restoration of damaged neurons and things like that. Yes, so that's about me, and how that really vision and research project basically turned into a company is when after doing UCL, I met my two other co-founders, Richard and Saran, and these guys are really into AI and have been doing AI and machine learning for decades. I don't know how long. And we kind of, they were looking into how to apply AI in the field of psychedelic drug development. And that's how we got together, that they would provide the expertise to develop the best AI drug discovery tools that would enable the development of the compounds that I've been working on. And that's what we've been doing for the past 2 years or so. Yeah.
It's such a fascinating story. There's so much in there. But before we can get into the idea of what a novel psychedelic looks like, we should probably back up and begin closer to your thesis. Like, so what was the thesis that you wrote when you were beginning your work at uni, or when you were finishing up, I guess?
Yes. So this is about basically looking into the mechanisms, how psychedelics act on the basically biochemical level, and they bind to different receptors within the brain, and the main psychedelic receptor for the classical psychedelics is the 5-HT2A receptor. And if you activate it in a certain way, then that somehow leads to psychedelic effects. And there are also other receptors that these compounds may bind to, and that also affects the overall effects they will have. And I have been, so basically if a drug targets more receptors at the same time, this is called polypharmacology. And it's usually these different polypharmacologies that make up the specific properties of a drug within a series. And I have decided that there was this one specific receptor that some psychedelics were binding to. And that's what I, studied, trying to understand what is the role of the second receptor for these unique class of compounds. And currently I can't talk about the second receptor yet. And that's because I have now really am in the mission to get patent protection around this, as unfortunately there's no way to get a drug to market without patenting it in this time and place. And this is something that we need to work around with when really going for this very, very ambitious goal of getting a drug actually approved through all the critical trials and so on. And so in order to secure that money, you need to play the patent game. Yeah, that's the hard truth.
Yeah, it is. It's super exciting to hear though. Like I speak to a lot of different people in the world of psychedelics, and you're the only person I've ever talked to who has explored this idea or this discovery of a second receptor that something can be binding to. And if you can't answer any of these, just let me know. Does it seem as if there's a firing pattern that leads to the psychedelic experience? Or, like sometimes like a spark plug would hit like the 1, 2, 4, 3 plug. So is there something in a firing range that leads to the experiment, or is it just the attachment to the receptors?
So from the scientific evidence on the very much lowest level, there seems to be a pattern that you can have a drug binding to this 5-HT2A receptor that's associated with the psychedelic effects, and you need the receptor to change its conformation to a different one, which is process called activation. But the activation actually is like a very much a spectrum of things. You have so normally this is a receptor for serotonin, and if that binds to it, that would be called the classical response and the natural response. But if you have slightly different compounds of serotonin that put the receptor in a slightly different shape, that still can lead the organism to think, okay, this receptor is activated, but it looks kind of different. So the response is also different from the body. And psychedelics are this weird class of compounds that produce a specific shape of that receptor that leads to these effects. But this is just the beginning, right?
Yeah.
When that happens, it basically kind of how I see it is that this new shape of the receptor upon binding to a psychedelic is interpreted by the body that it really doesn't know what to make of it, and it introduces a bit of a noise within the signaling pathways. Basically, like it can't read, is it a 0 or 1? Is it 0 or 1? I'm not sure. And each of the neurons maybe reacts in a different way, and it just introduces noise within the system. It's like there's the some of the signals are sent to wrong areas, and there's just a bit of a mess. And that probably is a very simple explanation of how these effects come together. And yeah, it's basically that the noise means to more firing of the neurons, and the signals getting to places where they wouldn't have normally traveled to, which is basically, this you get new perspectives, new associations, while at the same time the brain feels more connected overall because there's connections where there haven't previously been any, and from then on arise the psychedelic phenomena that we experience.
Man, it's so fascinating. Okay, so when we talk about the actual receptor site changing shape, is that confirmation that psychedelics do in fact change the structure of the brain?
This is very dynamic space.
Okay.
And at this level, where there's just molecules, and molecules change shape all the time within a cell.
Okay.
When we talk about changing the structure of the brain, more often that is talking about the neurons, the cells that are bound together in some such ways. And essentially how I imagine when we talk about brain circuits, for example, which is the brain structure, it's these neurons have axons and dendrites, which are the connections to others. And along those connections, they send their signals. And when you're sending a signal somewhere, the more you send it through the same connection, the connection grows and becomes stronger. And that is the process of learning. And that is just completely normal. But it can happen that we create a connection that is not working out for us, that is some kind of toxic loop of negative thought, right, or whatever. And when we use it often and often enough, it becomes so strong that we cannot really fire the neuron in anywhere else but in this direction of the negative thought, right? And here, psychedelics can help by temporarily allowing the neuron to brain anywhere to fire in other directions as well. And when it does, it actually creates new tiny connections to different places, which is a process called neuroplasticity, or more technically, it's dendritogenesis and synaptogenesis, so that there are tendrils growing, and then the tendrils connect to a different neuron, which is the synapse. So these new connections are made. And this allows effectively a novel avenue for thought, which is great. But obviously, these are, after one trip, kind of very small. And if we don't use them, they're gonna, disappear again. So that's why all of this integration process is super critical for retaining the benefits of psychedelics. And you need to send the signal through that new connection that you've made while tripping to make it grow and be a constant option that you can use with your brain later on.
It's really well said. And as you were explaining that in my mind, like I'm a huge fan of maps. I love maps. And like if you look at a big map and you look at a river system, you can see the main artery of the river going down, and there's all these little tributaries off of there. And the way you were explaining that, it seems to me that that's almost like a good metaphor for what's happening, because you're starting to see these little things going. And if you just said, okay, I'm going to dam the water up and shoot it down this bypass over here, you know, the same way people who are stroke victims learn to walk again, they got to teach themselves to do it again. And the way you describe the integration with psychedelics, it's so moving to me, because I know it's not a panacea, but I really am excited to hear about the research you're doing and hear that explained that way. If I may for a moment ask you this question, which is relevant, you know, it seems to me that long-term psychedelic use, like if you get on a program of using psychedelics, maybe you microdose, you know, 3 weeks in a row, and then on the 4th week, you do like a 6 or 7 gram dose. You know, it seems to me that you're also becoming familiar with the psychedelic environment. Like just like any environment that you explore, the more you explore that environment, the more you become aware of your surroundings in that environment. So my question is, do you think that there is something beneficial about getting to understand the psychedelic environment, not only for you, but for helping other people who may visit that environment later?
Mm-hmm. Yes, I think you're getting into the realm of how do we work with the experience while we are in it. Yes. So I have this other metaphor that I'm sometimes using, and it's often when I'm talking about the concept of cognitive liberty, is that you have this basic sober mind state, and that is essentially one reality. And you can change their reality by walking over to a different place within the physical world, which changes where you are. But you can also change the reality by modifying the way your brain processes the signals from where you are. And each drug may basically elicits this unique change in the way your brain processes information, and that in itself could in some sense called be a new place, and each of this drug now has a new place. And it's about yes, if you're new there, you're probably going to be a bit lost. You know, like what's going on, where am I, what's this? And you will need to familiarize yourself with all the ways how what you're normally used to gets processed in this new mind state, because it's unexpected. You don't know how it's going to change it. It's not natural. So in this sense, you can learn and you can see, okay, like if I look at this white wall, I'm gonna see patterns. It's just, you know, simple as that. But there's a lot of these things that people who have never been in this state will not be familiar with. And if I go to the fact that it can help someone to familiarize themselves with it, I think it's important for this therapeutic aspect, because when you initially have this experience and you're in this new place, you're mostly going to be filled with wonder and awe and these philosophical questions, what about the nature of the reality and so on. And this is kind of good to think about, but there's usually no real answers to, to that. You could think about that your whole life and
Yeah.
not get anywhere. So in that sense, it's good if you pass that point, you get familiar with it, and then you actually use it for more immediate needs for the therapy that you want to focus on or something. Uh, I see it especially often with DMT, where people report the first experiences like, wow, how is this possible? My brain cannot do that. That's not possible. My brain has generated these crazy, 3D, full HD patterns or visions. And then only after a while you can get to notice that there's some things from your subconsciousness appearing within the vortex of visuals that can give you clues on what you are not focusing your attention enough on, or what you should be doing, and things like that you kind of only get to pick out when it's your I don't know, third, fourth, fifth session.
Yeah, you got to be careful, or there's death by astonishment. You just you're like, I can't, and then it's gone.
Yeah, exactly. So fast, also, that you don't even have time to
Right.
think about it, all of it. It just.
so that is an incredible way that I think about that a lot. I think about the landscape. I think about the intention of going to that space and doing my best to be present there. However, I juxtapose that with this other idea of and this is, I've heard it explained in a different way. And I think that they're kind of at odds. And this other way that's explained is that psychedelics, they decouple memory from experience. And so it's almost, this other idea is that, maybe it's not so much novel as it is erasing the current layer of memory that you've tied to that thing for the last 30 years. I don't know if those are at odds or if they work together, but what do you think?
This is super interesting. I believe that a lot of therapeutic value that we can get is exactly as you say, take our memories and reevaluate them in a different light. And that's definitely what psychedelics allow you to do. And I think it's because you have so I don't know when it starts forming when you're 4 years old, but you create this operating system within your brain. And that really, people don't realize how many things are encoded in that system. What are all the minute things that you learn when you're growing up, and be it in the speech and interactions with other people, in how you tie your shoelaces and all of that. And this is really, and when you process new information, you really always check with the settings of your operating system whether that fits in or not, and you always see them in the perspective of what you've learned before. And you can have some, for example, memories of trauma that you were looking with one perspective that you had at that point when you experienced that. And that was like really, really horrible. And you cannot really solve that with the mindset that you had before, but it's encoded in the language of the mindset. And even if you grow older and your mindset overall changes, you still have this old memory there that's, part of a old software that's not been updated for years and years. And you need to take that out and kind of convert it into the new one. and see it from the new perspectives of the person that you are now, where you have ideally the input of a therapist who are the best source of how to call it, functional and efficient operating systems of the brain, where they can really kind of fix the bugs that you might have. But I'm gonna stop this line of metaphor.
I think it's a great metaphor. It's difficult to try and describe a landscape where words fail. And I think in the psychedelic experience, words begin to fail. So many people find yourself deep in this trip experience, alternative realm, however you want to explain it. And while we can use words to describe it, it's very difficult to explain it. I can say that there's this alien that is coming and talking to me, but that doesn't really describe what's actually happening. You can just get as close as you can to it. And it may be possible that metaphors, analogies, even poetry is in fact the proper way to speak. That may be the only way we can really connect to each other, is to use that type of language. And I'm wondering, what do you think is the connection? It seems to me that there is a really big connection between psychedelics and language. In your ideas, what do you think is the connection between psychedelics and language?
So you've actually said it very well, that language is essential for communication between two people. So one person has this experience that is, multidimensional and super, super complex, but even without any psychedelics, yes. So like what you feel and what you perceive, that is like way, way, way more data than what you cannot directly transfer it to another person. So you need to compress it within language symbols, and the other person has to read these symbols and extrapolate from that something that you've tried to get across. And it's obviously not going to be perfect. It's basically a compression where a lot of the information is lost. And I think we've done a pretty good job developing our own language systems as humans, and it allows us to, work together. It's what connects us. So I guess there's two aspects. First is under psychedelics in the acute experience, the words break down because of the chaos within the brain. And then when we are, sober and can integrate it, we still don't find the words, but that's because we cannot find the words to describe that. So I guess there's two aspects how language fails here. And I think this is very tricky in terms of how to convey to people that there's actual value in these compounds. And I think that this plays a massive role in terms of prohibition, and how to approach that is that the inability to come close to a description using words for the people that are the decision makers but have not had the experience themselves. And really there's this other world where you not even barely aware of what's there. And so I see a problem there. And another problem with being unable to use good language to express it is in terms of the development of compounds that are supposed to have a certain effect, therapeutic effect, And when you cannot even describe that well in words what the compounds should do, then it's very difficult to try and develop something like that. So it's definitely in the early phases, it's more of a trial and error, following some more basic information about these compounds, but the way how they interact with the human brain and what are the nuances of the experience that we cannot really well describe with words, even though we're trying. That is a long way to go, from chemical structure of the molecule to these nuances in the trip, or like what kind of colors are present within the trip, or things like that. That's just such a long way. Yeah.
It's there's so much there. I love the way you explained it. I do think that this issue of language is something that humankind has struggled with since the Tower of Babel. However, it's so fascinating to see it come to a head right now, where in the world of clinical trials, scientists want to see objective results, but so much of the therapy is subjective. So much is trying to explain an experience, and there's almost no way to quantify it. So, even when you can show results and show a patient who may have gotten over PTSD in 1 or 2 sessions, how do you measure that? Like it's almost to a point where the people writing the check don't have anything to go on except the words of the therapist and the patient. It's a conundrum, right?
Mm-hmm. Yeah. So there is, I guess, I'm not sure that I completely agree with you that it's impossible, in the sense that this is true also for other forms of psychotherapy and interventions in the mental health sector. So this is something that we're struggling with on other fronts as well, not only in the psychedelic world. I think that what the psychedelics bring in terms of uncertainty is one, of course, the placebo effect, that you cannot control within the properly within clinical trials using psychedelics. And I think we're trying to get okay, we don't really care that the patient has to self-report what has happened to him, because that's what we do in all the other indications. That's normal. But in terms of what we ask the patient going through that therapy is really important, and the question is whether we are asking the right things, so that the person maybe having his experience for the first time can understand what we mean. For example, like what happened within the session, and I think that we don't have the language developed for the individual things that happen when we are within the changed state of mind. And I really like some of the efforts. So, for example, people from Psychonaut Wiki, they're developing this subjective effects index, which is a collection of all the different phenomena someone is experiencing and under the effects of psychedelic drugs, and categorizing them and naming them, basically. And I think there have also been some on DMT Nexus and some forums. And really, it's super important that we go to this place of of altered state of consciousness often, so that we, familiarize ourselves with it. And once we start to develop language for things that happen to all the people in common, we can have a much more meaningful discussions, or we can begin to talk about these things. And I think that we're a long way off before we can really have, I don't know, meaningful discussions about closed eye visual experiences of like high dose psilocybin, where we see, I've seen this phenomenon and that one, and yeah, I've seen that, last week, and
Yeah.
uh, that's kind of quite out there. People are usually just describing it in their own words, and uh, then it's like you get the feeling that it was something amazing, but I don't think we're able to share the content of these experiences effectively yet. I think we can improve. To what level? I don't know.
It's brilliant. It's brilliant to think. It's almost like we're building a new alphabet so that we can have a conversation. We're seeing these things for the first time, and it's really fun and interesting to get to talk to someone who is so knowledgeable and is creating, in some ways, a roadmap for people to follow, and in other ways, which we should maybe can shift gears and talk about, what does it mean to begin drug designing a new novel compound, perhaps a new type of psychedelic? Can you run us through that?
So now the drug design process is mostly done in silico and computers,
Mm-hmm.
and it's about describing the relationship between the chemical structure of a molecule and its biological activity or its effect when administered to humans. So, as you imagine, this is like really, really complex equation with a lot of variables coming into that. And you need to take into account, so the most crucial things that are driving the activity of a compound is, as I described previously, how it binds to the receptor and what kind of conformation of the receptor it creates. And given the fact that there's, tens of thousands of different receptors within the body, and all the other things that all the different systems that we have, then yeah, you need to take all of that into account. But how we simplify things is that we just focus, or firstly focus on the receptor or receptors that we want to be the drivers of that biological activity, and we model how these compounds bind to those receptors. And that can be done either through like learning the patterns of previous drugs, or actually modeling the structures of the receptor and trying to fit the compounds within the receptor and see how well they fit. And this is the early design stage where you're really trying to look for what possible molecules might have that activity. And then it's the much more tedious, longer, and cost efficient, or like cost intensive process of actually testing whether that compound works. And we need to go very small like in vitro assays to see really whether there's an affinity between those two compounds, what is the pathway that it gets activated when these compounds bind, then does it have effects on larger structures like cell cultures, then organs or smaller animals, and then eventually humans. And that's also a long process still before we can proclaim, okay, this is safe and effective to be used for treatment. So that's just the general overview. Obviously, there are so many parts that go into that that probably it would be best if there's something within that process that you're most interested in that I can talk in more detail about.
Yeah, it's such a giant thing. And as you're explaining it, I'm thinking, wow, how would you even form a model? I know like on mice, you could crack their heads open and like look in there and stuff, but obviously for human beings, like that's a little bit unethical. And so, you know, I guess maybe I'm not even sure where to take it from there, but I guess I'm wondering like, in today's world, we can use EEGs or EKGs to kind of see a little bit what's happening there. But where are we right now in the ability to monitor what's actually happening in the brain on a psychedelic?
Mm-hmm, mm-hmm. You are hitting the later stages of what we would normally consider to be the drug development process. And so I know that you have spoken to Nick Murray, who
Yes,
is
I love that guy.
who is much, much more knowledgeable in this than I am. It's about I suppose monitoring as much as we can throughout the entire body.
Mm.
And for brain, you have functional magnetic resonance imaging, which is super expensive, and you have to, be inside of a huge magnet to get the readings out, which is, I guess, the most sophisticated way of looking within the human brain in real time. And but I've heard in our accelerator that we were part of that there was a startup trying to get fMRI working even with much smaller magnets than are usually required. And really, I think that it will be possible through new technology to make functional magnetic resonance imaging much smaller, much more compatible and accessible. So I think there's still a long way to go, but right now, as you said, we can do EEG, then fMRI, and then what Kernel is doing is monitoring the flow of oxygenated blood, basically within the brain. And I see this as not how traditional drug development process usually goes, for whatever whatever people may say. It's very much about trial and error still, and we in a lot of drugs, and you may be surprised, we just don't know really how they work and what kind of effect they have. But if you design clinical trial that looks at some outcome that might be very abstract, as you said, in mental health treatment, we're just asking people a bunch of questions, and depending on what they say, we determine, okay, that was a success, that wasn't. And what happens within the body to cause this response, we often do not know. And in much simpler drugs, so doesn't have to be psychedelics to
Right.
be approved, but we still don't know actually how it works. And I think that, yeah, the psychedelic research is mostly within that phase that we're, or if I'm talking about novel psychedelic
Hmm.
research, we're just trying out. And this is also because this is a chicken and egg problem. You would ideally want to run all these different brain scans under the influence of a certain new molecule before even, spending a lot of time of developing it and
Right.
proving that it is safe and effective. But in the current environment, you would anyway still need to, bet on some drug and put a lot of money into it to develop into its stage before the regulators even allow you to give it to humans to actually make that brain recording. Um, Unless you're Alexander Shulgin, of course, then you.
we
Yeah, yeah.
Beautiful. I when I think of novel drug design, I think that almost the entirety, with the exception of Shulgin, with almost the entirety of drug design is an exercise in like confirmation bias. Like this drug does this. How do you know? Because it seems to do it. Like the best guess. It's just the best guess. But here's what, and tell me if you think this is crazy. I think that the okay, it's tough to get this out. I think that you can theorize what's happening in the brain better than you can demonstrate it on any kind of machine, EEG, EKG, and even when you use those machines, all you're doing is measuring confirmation bias. But I truly think, you can go back. Okay, you can go back to like if you read Plato's works and you read Socrates and Timaeus, there's this one scene where Socrates goes into the dirt and he takes this slave boy who doesn't speak. They call him a barbarian because he's not a barbarian, but it's Babel, right? And he takes this young boy over here and he draws like a square, and then that guy draws a line through it, and he's showing everyone like, look, here's a guy who thoroughly understands the dimension of this cube right here. I didn't have to tell him anything. He just drew the line there because he knows what I'm talking about. And when you see things, when you read language like that, when you read stories from the past, when you take a psychedelic and you see a 3D object spinning in front of you, like I think that what we're seeing are the solutions to the problems. Like if you can see the same tetrahedron moving in front of you right there, and you see it and I see it, like maybe what we're seeing is the activity in the brain right in front of us. And if you can go into the psychedelic realm with the intention of understanding neuroplasticity, I think you can see what's happening in the brain. And I think that's just as good of a drug design as measuring it through waves or measuring it through some sort of EEG or oxygen in the brain. Like, I know I'm kind of way out. I'm just way out on left field right now talking about this, but it's plausible, right?
Yeah, 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 yeah. I think I understand what you mean, and the thing that is the thing why people are not using that more is, I think, just because this is unique to psychedelics. As if you were to use this in the design of, for example, I don't know, cholesterol reducing drug, you're not. You're not gonna see the cholesterol being broken down by by some process within your body when you take that, and this manifestation of the effects of a drug that we can directly perceive, as you said, in front of us, is something very, very unique in the sense we of course we have that with I guess other drugs also have very tangible effects, and that would be possible to use your own, let's say, um, organism to try and, um, you know, see whether it is active and what effects there are. But why that, and I'm getting to the main reason why this is not used, then is because essentially you are, without a proper understanding of the molecule on other levels, you cannot really predict what it's going to do, because even tiny changes to the molecular structure can have really dramatic effects on the activity of the molecule. And so, for example, you have a series of compounds, and you know anecdotally some of them are psychedelic. So you just assume that, okay, this drug is also going to be a psychedelic in a similar way, and it's not going to be a neurotoxin that's going to, uh, kill my brain cells.
Right.
And this is the fact that it is inherently risky to do this self-assessment of a drug, and that's why the regulating process it doesn't want people to, do that. It is. I think there are different rules for actual self-administration. I think in some ways in some countries, it was accepted. I think there were even some articles beside Shulgin's work where people have experimented with new drugs on them and reported that in the scientific literature. I think in Germany it's allowed ethically as long as it's the. But I mean, I might be lying there, but I think I remember something like that, that as long as it's yourself as the researcher, you can do it on yourself, on your own responsibility. But in this world, it will still not give you enough data anyway, because for anything to be reliable, you need um, uh, more measurements, more people to do that. And it is inherently, taking on the risk of trying a new drug onto the humans, and it's probably difficult to make sure that the people you give it to fully understand the extent of the risk that they would be undertaking. So that's why these mechanisms are in place, that when clinical trials for any drug are done, that it's, first of all, there's enough preclinical evidence that there shouldn't be any kind of toxic effects. And then that, all of the people within the trial are unbiased and know what the risks are and so on. But there's this question that is really something to think about, and that is, we have basically outsourced the risk from humans to other animals, and that's what makes up the most part of the preclinical class, or not the most part, but historically it has been an inseparable or necessary part, is that you test these compounds on a lot of different animals, smaller first, then even larger ones, to observe the toxicity and kind of judge whether or extrapolate whether effects seen in the animals might not translate to some toxicity in humans. And obviously, there's people who are rightly concerned that we are just, taking the risk and suffering onto animals, and so that we can, be safe. And this is like very, very difficult topic for basically ethics and philosophy, right?
Yeah.
And I like that we are now moving towards uh, through technology, we are able to actually do a lot of advanced systems in vitro. So, for example, we can grow organoids, which are like tiny organs in the testing tube or wherever, where you can even. So how does it work? Is that you basically take a human cell, and you can even take it from the specific human that you want to treat, but usually from the person that maybe has the disease, and you can reprogram it to revert back to, the state where it can change into any kind of cell. And then you can tell it, okay, now grow me neurons. And it starts to grow neurons. And if you provide the correct growth factors, it will continue to grow into a neuronal culture that is going to differentiate within uh, into basically its own an entire miniature of a brain, and these are called cerebral organoids. And this is a much better model of actual stuff that's going on within the human body, and it's also much better translatable to humans because it's actually human cells and human receptors. Because you have this problem when you test some toxicity of a compound on mice, it might be toxic there, but not toxic in humans, and vice versa. So there's because we have different structures of the receptors, different sequences, different sets of genes, and so on. So there's these advances in organoids have actually resulted in, I think it was NDA or some of the regulatory authority accepting basically investigational new drug applications, which is an application for whether you can administer that to humans. And you can now get that without any animal studies, just by doing that. And it's also sometimes supported by microdosing, which is actually an equivalent of what Shulgin was doing, but you can usually you need like a body of preclinical evidence on these in vitro models and organoids. Then you can get microdose study approved, where you put like very, very tiny amount of compound to humans. And then you can actually, if nothing happens, nothing bad, then you can go on to phase 1 trials with the full dose of the drug.
Man, that makes up so many questions in my mind. Have you talked to the Shulgin Institute? Have you talked to those guys over there?
Yes. We've had one call with them some time back. Yeah.
Man, he's got books, and I got the PCAL and the TCAL. Like, he just has books of molecules that he's made, and like, okay, but this brings up another question. Why do we need novel psychedelics? Like, why do we need them? Like, maybe we just play around with the dose of the ones we already have, and maybe different doses of psilocybin cure different types of mental illness. Maybe we already have it.
Mm-hmm. Yeah. That's a very good question, and I think that the drugs that we have are super valuable, the classical psychedelics that we have now. And their value is really in the mental health world, where we have found very efficient ways how to, do the psychedelic assisted psychotherapy and get meaningful results using that. And it's also, very safe. But you have to, from the perspective of general drug design, you usually how it goes is that you have these initial lead compounds that are sometimes called first in class, that are oftentimes derived from nature, or they're with the case of psychedelics, you have some serendipitous discoveries like LSD, and this is just something that we have like found, and we applied the therapeutic process onto that. And in that sense, the therapeutic process and the therapy has to adapt to the properties of the compounds that initially we found that has these effects. And when we see that this is working, and this is called like validation process, basically, we see that happening with psilocybin for depression, for example. And this tells us that, okay, we can use drugs that bind to this 5-HT2A target to use as a basis of a treatment for mental health disorders. But then you ask yourselves, okay, and if we want to treat that, could there be a different drug that would in turn be optimized specifically to be used in this treatment? And then you have the option to mess around with the properties of the drug to better actually fit the needs of the treatment. And I think that is always the phase in drug discovery and development. For example, with penicillin. So you have this, and its antibiotic properties is awesome, but okay, it doesn't work on these guys, then it doesn't work with these organisms. So okay, we're gonna tweak the structure, and now we're getting the better antibiotics. And penicillin itself is hardly ever used now after all those years, because we have all these other range of options that turned out to be, and statistically it's very likely because the possible space of molecules is so vast that you're probably not going to get the perfect thing for specific treatment in one shot. And when I come back to psychedelics and the first generation of them, there are several ways how these could be improved. So one of the limitations is that the treatment of high dose psychedelic is demanding and is contraindicated for a lot of patients, and it might actually be also a bit of risk to individuals, for example, that are old and have some comorbid diseases and so on. And it's a question how to make it available for people that would not pass the requirements to be accepted into psychedelic-assisted psychotherapy. And then also, for example, what we're focusing on is that there is possibility of using psychedelics for neurodegeneration and traumatic brain injuries. But this, as you said, is very much a different paradigm than the psychedelic-assisted psychotherapy, where you take one big dose once in a couple months. You need to, regularly dose that so that you continually have the effect of these drugs. And that is very difficult with their tolerance buildup. And there is also a risk of cardiotoxicity because these drugs bind to 5-HT2B receptor that has been associated previously with cardiac valvulopathy and development of heart problems when administered regularly. And so we are aiming with April 19 to actually solve all of these problems at once. And it's actually by introducing this novel target, we have compounds that are inducing neuroplasticity and additionally also are neuroprotective, and still have the same mechanism of action, but supplemented by a different pathway also. And this removes the reliance for the effects on neuroplasticity to be tied with this 5-HT2A target, and that allows even in lower doses to still have a potent neuroplastic effect. And there is space for actually looking at different compounds and seeing whether their combination of a duration of effect and the tolerance that they induce and the I want to say cognitive impairment, but this is something that we cannot really model now. It's obviously going to come into play at later stages. But we, and also 5-HT2B binding, which is corresponded to that cardiotoxicity. And those are just the main parameters of a drug that we can fine tune early in the process. And we can find a compound that would be safe and effective, even while you would be regularly dosing these elderly and comorbid patients that have, neurodegeneration or that have gone through a stroke or some other brain injury, traumatic brain injury. And I think that is something that the first generation of psychedelics does not address well. And really, if there was a drug that I'm describing and we think we can make that, then this would really open up the possibility of applying psychedelics also in other areas such as mental health. And yes, I think that there might even be improvements in mental health, in making some drugs more safer, last for a shorter time to afford, get more affordable therapies. But I think that the possible for innovation is, as you mentioned, kind of small, because we already have super powerful drugs that are super effective in mental health. And that's why we as a company are not focused that much into that space, even though there might be applications of our compounds within mental health also down the line. But what I really would want to do is that, okay, we have discovered this amazing mechanism that produces, higher neuroplasticity in the brain, and this allows for the neurons to, as you mentioned, in stroke, some parts of your brain die out, and you can, by inducing greater neuroplasticity, tell the surviving neurons to, connect in such ways that they actually replace and restore the lost functions of those neurons that have died. And this is very much what psychedelics do. This is what I talked about earlier, creating more connections within the brain, more synapses. And we have very limited data to support that psychedelics will be effective in neurodegeneration. But I think it's a lot because people have been mostly focused on really proving to all those, politicians and decision makers that these, compounds have value. And the best way is to do that in mental health. And that's why, and the research was limited and kind of hard to get approved. So that's why not many people were really ventured into the neurodegenerative area. But I am super convinced that it's going to have a great effect. Not that it would be disease modifying, that it would cure, Alzheimer's or things like that. That's not really very likely, honestly. But we don't have cures for neurodegenerative disorders of this magnitude. And what we do anyway is symptomatic treatment, which has a lot of side effects. And then we have supportive treatments of some sorts that some look kind of promising. But for a long time, I believe the best thing we can get is focus on supportive treatments, and psychedelics are perfect for that. Also because they approach the problem, even though they might not cure the cause of the problem, they approach it holistically. So when you have a patient with neurodegenerative disorder, they will, be freaked out. They will have anxiety, depression, and all these other associated
Yes.
mental health issues. And if you can, have a compound that will address both your mental state and the state of your brain, the neurology, I think that's going to be a very powerful tool in helping these patients to, keep their dignity and within this, very, very horrible state that is neurodegeneration and dementia.
That is that's so awesome, man. I love every part of it. And there's a few things that I would like to address. Number one, I think you could say that depression and anxiety may be the warning signs of Alzheimer's before it happens. The same way someone loses movement in their arms when they have a stroke, or that there's telltale signs of things that happen before the actual big event happens, whether it's on the planet or whether it's in your body. And maybe if we can use the psychedelics to address the anxiety, the depression, maybe it's like getting to the Alzheimer's before it happens. Maybe that's where the magic is. I couldn't agree more that they are going to see psychedelics, and this is just an opinion. I'm not a doctor. I'm a truck driver, but it's just, I wholeheartedly believe that you're going to see the use of psychedelics in treating neurodegenerative diseases. Where I think there is going to be an issue, and DARPA is actually doing a study on this right now, where they anesthetize people and then give them the psychedelic to see if it has the same kind of effects. I'm of the opinion that you have to be conscious. You're going to have to do the work. Like you need to see the plants breathing. You need to face the demon, whatever the demon is. Maybe that demon manifests as you thinking you're an alien, but whatever is going on in there, I think there's a component of consciousness that needs to be active while it's happening. I could be wrong. And maybe that's why DARPA's pumping $30 million into this study. And it didn't even make sense to me until you started bringing up the idea of traumatic brain problems or neurodegenerative diseases. And like all of a sudden I was like, oh, on the battlefield, Alzheimer's disease. What's the biggest plague coming our way with all these baby boomers retiring? Like, they're really, even though when you talk to people like Rick Strassman or all these geniuses that came before us, and they say, listen, we've been here before. It's not a panacea. It's so easy to start to see what it could be used for. And I'm just so thankful that we're at a time where we can begin to do it. And I'm so excited to talk to you, man. This is amazing.
Yeah. And you touched up on a very important issue that a lot of people are warning about, and they're finding very, I would say, controversial ways how to deal with that, and that's the aging of the population.
Yes.
Right. So our birth rates are dropping, and this might be maybe a bit of a conspirational theory, but we've actually seen a lot of this pro-life and anti-abortion movements. And I mean, I can't help to associate it with the drive of the governments to, get people to make more babies so that they're not put, because they see that we're dangerously approaching the point when people are just going to be aging, and there's going to be a lot of old people, and the demographics are going to be maybe unable to support the entire civilization.
Yep.
Because those old people are going to require all this kind of care. And now you have also longevity companies and researchers trying to, extend the human lifespan even more. And what I think should be done in this longevity is really, really to focus more on the health span, on actually making sure that the people are healthy when they age, not how long they will live per se. I mean, that might be as an afterthought, or just a secondary thing that you want to solve. And for example, dementia case caused by neurodegeneration or other means is really at this point, once you're older than 60, your risk of dementia exponentially increases. And there's been studies that it actually continues well. You know, it gets worse and worse. And so there was this study of people aged over 90 years, and about 42% of them already had dementia in some form. And then, you know, the incidence was still rising exponentially to of your risk to get dementia. So if we don't solve that, we're going to have a lot of old people, and they're going to require a lot of care, because this is so intensive to care about someone who has dementia. They're slowly losing touch with the reality,
Yep.
losing their independence, and it's actually horrible. So I have also bit more personal experience seeing how the people are slowly, you know, losing it, and it's very scary, and not only for the person themselves, which I cannot even imagine, but for all the people around them that need to, you know, be with them in their difficult states, and you know, help them do basic things. Yeah, so that is something that I really want to address. And I see a big potential in using psychedelics for this. And you said that yes, there is an approach in trying to say, okay, like we don't need this psychedelic effects for this class of compounds at all. And I also disagree with that. I think that yes, there would be an effect, but what is this, you know, selling point of the psychedelic drugs is that they actually do this chaos within the brain that causes inspires the brain to, you know, find other ways of connecting together, and start this rehealing process. And I've also used this metaphor about psychedelics, macrodose of psilocybin. It's okay, like you're exploding the house that is your brain, and then, you know, you're slowly, you know, building it up again throughout that experience. And that is most effective, you know, because then you would have the non-hallucinogenic or non-psychedelic psychedelics that, you know, you still have the brain, but you're just trying to build some things on top of it, you know. So that's obviously not as effective to if there's a broken house trying to just build over it. And what we will be aiming for, and this is obviously very abstract, is to retain that element of breakdown of this entropy increase, but do the demolishing of that in a more controlled way, that the people could actually really be in control, but see that, you know, that there is some way of and this corresponds to the visions that you said, that is the chaos and you know, kind of destabilizing of those previous neural circuits. But you ideally would want to have it in a way that is not preventing you from functioning in, if you want to use that in this neurodegeneration and traumatic brain injury treatment. And so that's why what we are aiming for is I would call non-intoxicating effects of these psychedelics, as opposed to, you know, non-psychedelic or non-hallucinogenic. And I think that there's a way, there's a threshold, even when doing psychedelics as a microdose, the current psychedelics. But I think that it's not enough that you're just taking a microdose, because there's this rapid onset of tolerance. You cannot take it continually, and there really is not enough of an effect as we're getting from various studies. to actually have a therapeutic, really significant benefit. It can have effects on people's, creativity and ways of thinking, and there's definitely effect for that. But if you consider that your brain is degrading at a rate that no other drugs that we try to develop over 100 years is able to stop that. The results that we're getting for microdosing with the current drugs, it's very unlikely that it could affect that to a significant degree, in my opinion. I might be wrong, but we don't have that data for humans yet.
It's fascinating to me. I did a self experimentation where I did like between a half a gram and a gram a day for 30 days. And I was surprised, and this is just my own subjective results, but I was surprised at how little tolerance that I actually felt. If you read James Fadiman, or you do your research online, you can see that the average microdosing procedure is probably like 3 days off and 1 day on, or 2 days off and 1 day on. But I did find the realm of creativity was easily accessible. I did find that the colors and everything was turned on, if that kind of makes sense. And it seemed to me, depending on how much sleep I got and my diet, but the feeling was always there. But I want to shift gears and go back to the idea of getting the neuroplasticity and potentially having good or dramatic effects when you're in an unconscious state. So you had said that, like, at the high doses of psilocybin, like, there's the explosion, the chaos happens in your brain. And then earlier in our conversation, we had talked about making sense of the environment. Isn't the making sense of the chaos where the actual integration is? And if you are unable to be conscious, if you're unable to make sense of the chaos, then you're not getting the benefits. If you just have the chaos in your brain, maybe you have those tributaries or the neurons firing off and making new paths, but is that enough to get the changes you need for neurodegenerative disease? Like, I think I'm just trying to make the case again for you have to be in the realm of the big explosions and make sense of it in order to do the learning.
Yes, very, very interesting thing to think about. I believe that on some level, it's going to be slightly effective, even if you even if you are unconscious.
When you say slightly, I'm sorry. When you say slightly, what is that, like a 5%? Or, I mean, I know we're just kind of guesstimating. But when you say slightly, like, what would you mean by
that? So that's actually why we are introducing this other target into the equation, because there are other ways how to affect the unconscious pathways that are contributing to the survival of neurons, the neuroprotection and neuroplasticity, and there are other drugs that induce neuroplasticity. For example, a range of antidepressants, but other drug classes as well that help this process. So definitely, there are ways how to, like, if we're talking about the neurodegenerative indications, then this would have some effect. I don't know about the percentage. I can't guess, but what is, I think, shouldn't be lost is that as your brain degrades in that state, you will lose some of the neurons that have been doing some functions, right? And that's why you're like stopping to function properly in some aspects. And one thing is to just slow down the degradation process, and that you can do, and that's okay. But even on subconscious levels and subconscious mechanisms, if you want to call it that. But I think there is a lot of benefit if you were to be in this neuroplastic state induced by psychedelics that introduce a little of chaos, but you can still function, where you can find more efficient ways of performing the functions that you need to perform, even with the limited amount of brain cells remaining. And that is very much about actually learning. It's like, so you are when some connection in your brain dies out, you will need to find a way how to work around that. And psychedelics, if you have that state of the neuroplasticity, can allow you to or maybe also work with input from some therapist or a doctor to find a different way and maintain the functioning through alternative neural pathways. And I think that this is the restoration of function of the brain, basically. So that I think is very crucial and very beneficial to have what you're talking about, this sense of the psychedelic state that is giving you new ideas, new associations, new perspectives on how things could be done. So yes, I think that if you separate it into this neuroprotective part and then the neurorestorative part or function restoring property, then you probably need it for the function restoration to work to its maximum. But I'm talking here, I'm not talking about mental health. It's different in mental health. I think mental health treatments without this effect would be much, much less useful. And I just compare it to taking normal antidepressants. Is like, yes, you're feeling a bit better and you might be able to find ways of thinking differently, but there's no assistance in that. And it will be pretty much the same doing any kind of other talk therapy to change that, because your brain is neuroplastic on its own, right? But this psychedelic effects of actual, chaos within the brain, that is what's, the main driver, allowing much more profound change. And it's about, I think, this very important aspect is that once you are, if you're in a normal state, you can only make such changes to your thinking that you can imagine. You need to imagine that a different way of thinking is possible. And it is very hard job of a psychotherapist to explain to a person that they can think in a different way. They don't see it. They have a wall in there, right? And then you take a psychedelic and it shows you that there's actually, 10 different paths that way, and you can have chosen and you can choose that path during that experience and find that it's there. And then you can kind of, integrate that and really, change your way of thinking. And if you don't have that, those random associations running through your brain in that state, or you're not aware of them because you're drugged under an anesthetic, then there's no way to see those hidden doors that are there in your mind.
Man, I'm it seems that at least in the West, like when I think of the drug companies like Pfizer or all the drug companies over here, for so long we've used medicine as a coping strategy. Like, okay, I know you hate your life, I know you're depressed, I know you're anxiety, but just take this pill and now you can continue to do the shitty life that you're doing and you'll feel better. But psychedelics seems to offer a new method of like, this is not a coping strategy. This is you realizing that you have a real problem and you can fix it. And here's how you do it. Like that's real medicine. That's real work. And like, I'm so excited for the future. I'm so excited for what you guys are doing. Like this conversation has blown my mind. I am thankful. I know you're getting close on time, but I wanted to touch really quick on this idea that we may not even be able to get into it, but you guys are working a lot with artificial intelligence. And in the world today, we see things like chat GPT and automation, yada, 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 and brain chips. But it seems to me there's a race between biology and technology. And on one level, you have the Elon Musk brain chip. But on another level, you guys are building cerebral organoids, which you can test natural compounds on. So it seems to me like we're using AI to further the biological natural processes of healing. So it's just weird that it's weird and it's beautiful, but it seems like there's a race between biology and technology on some levels, but maybe it doesn't have to be. What do you think?
Yeah, so I think that this AI that we're developing is actually our attempt to mimic the biology, because that is so much older than us, so much more advanced. We don't understand what it is, but we have picked up pieces of how nature does things and how they work. And we have transformed them into this world of silicon, and virtually we can replicate them. And if you look at concepts such as evolution that has, generated super complex things, we can kind of, do that in a computer and also generate, all these various things through the process of crossover and mutation and selection of the fittest. And so that's some part of what we're doing, evolution machines. And then another thing we've borrowed from nature is the structure of the brain, and those are the neural networks that are changing the world right now. And it's just a simple concept in it, that you just have various nodes that each processes information from nodes passing it to it, which is like a neuron sending a signal to another neuron, and it just does some operation on it and then sends it onward. And this kind of mechanism allows you to learn things, to remember things, to generate new things. And so we're kind of building essentially another brain for ourselves to think with inside these computers. And that's how it complements the biology research, is that we can outsource some parts of our thinking because it's getting so complex. Like what we're looking at is crazy. And by building, some helpers that can do that for us, that's great. And we can outsource, various tasks for remembering and recalling things. And in the sense of looking for actual new molecules, this has been used for a long time because it's much more closer to molecular structure, is much more closer to kind of encodable language that you can feed into these machines as are other things. So you can actually teach a model to predict some structures, some properties of a structure without doing all these real world assays. So it's the predictive AI. You can also train a model to generate a set of molecules that have a set of properties, and that's the generative AI. And this is really like so efficient now that medicinal chemists are slowly just losing the I guess edge of being able to perform better than machines at this drug design, and actually the best way as it was designed to get medicinal chemist and, give him a couple other brains that he can work with. And some of them, could be his lab assistant, but some of them are just computer brains. And by using them in smart ways, we can get so much more done so much more quickly. And that's what we're working on. But there's no time to get into the details, but maybe in another chat.
Of course. Of course. Where can people find you? And you got anything coming up, and where can people find you?
Okay, so you can find us on april19.ai. That's our website. But we're active on LinkedIn as April 19 Discovery, on Twitter as well, April 19 Discovery. And we are now trying to get money to run the next stage of our research. So to run all those real world experiments with the compounds that I've been designing and synthesizing, and those that are coming out of the AI tools and models that we've designed. We just now need to actually go to the lab and do the experiments. So definitely, if anyone wants to help us with that, they can reach out. And we're also open to collaborations. We're working with some universities and research institutions and other companies. So yes, we're open to discussions to push this thing forward.
That's beautiful. If you're listening to this and you are a fan of psychedelics, Tim Ferriss, if you're listening to this is the guy right here, Tim Ferriss. This is the next John Hopkins right here. If you're listening to this, reach out to them, check them out. These guys are on fire. It's such a fascinating conversation. I'm excited you guys are out there, and we'll get together again soon, and we'll have some more chats. I really enjoyed this. And
I enjoyed it as well. It was great. It was great. We got into some very deep and interesting stuff with you. Yeah.
It's fascinating. I'll let you go. I know you got to leave. I would talk to you for another 2 hours if I could, but I'll be in touch, man. Ladies and gentlemen, thanks for listening. All right.
Thank you. Bye.
Aloha.